part in this interesting new format for conferences. It's nice. I'm going to be talking today about our work using network-targeted brain stimulation to test mechanisms for resilience and aging. And even though the title is cognitive resilience, I'm going to be focusing almost entirely on episodic memory because that's primarily what I study. So for all of you who don't care about episodic memory, you can take off now. Sorry. So I think we're all familiar with the idea that many cognitive functions, including episodic memory, unfortunately decline on average across the adult lifespan. Either you're familiar with the data or you're just like me and starting to actually experience this phenomenon for yourself. Either way, it's a pretty robust effect that you can observe in most people. Of course, there's a lot of variability. Not everyone declines, and that's the part of the focus of this entire lecture series and a little bit of in my talk. How do we keep ourselves from declining, and what are the mechanisms by which we can experience resilience in the face of the normal ravages of age? So I want to start off with kind of an informative example from my lab and the studies that we run. So just to give you an idea, here's a typical MRI scan from a young, healthy adult picked pretty much at random from the subjects that we run. And here is a fairly typical healthy older adult. So this is a 74-year-old male, does not have any indication of Alzheimer's disease or mild cognitive impairment, no neurological psychiatric problems. And you can see just by taking a quick look, the marked changes in this brain versus the young adult brain. There's a lot of extra space in this brain, particularly around the ventricles, and the hippocampi appear to be somewhat shrunken in this individual. And just looking at this brain, relative to the young healthy adult, my gut reaction is just to think, aging. So this is a pretty typical older adult brain that we experience, So to give you a sense of the kinds of tasks that we use in our experiments, here's a particular memory task that's very challenging. And so what people do in this memory task, I'll talk about it a little bit later on too, is they have to memorize that particular objects like this somewhat dated video game controller and this giraffe go in particular locations on the screen. So they learn this association between an object and one of 6 possible locations. And they learn a lot of these things, up to about 120 of them over the course of several experimental sessions. And after a delay, after studying big sets of these, they have to recall the specific object locations where things were studied. And so it's a very difficult associative memory task. Young adults perform on average about 55% correct in this task, and chance performance is 1 out of 6. So they're well above chance, but still, plenty of room for improvement. And so what I'd like you to do is guess how participant F07, the older adult that I just showed you, did on this particular task. And so we're going to open up a poll. So again, take a look at the brain there, and you can respond in a poll, which I think should be opening up to see how you think he did in the task, either in the, there we go, 0 to 20, 20 to 40, 40 to 60, 60 to 80, or 80 to 100. And keeping in mind that young adults perform 55% correct on this task. And so go ahead and give it a guess. What do you think this subject did? I'm going to guess as well. We'll wait a few seconds for the poll to get completed. Michael, I don't know if you could let me know when you feel like you've gotten a good number of responses in.
Yeah, about 70% voted so far.
Okay.
Let's give it a few more seconds. Okay, so we're gonna end the poll.
Okay, what was the poll response?
Share the results. So
aha! Okay, 40% of people thought he would score in the, I don't know if everyone can see these poll results, but most people picked 20 to 40 with a few people in the other categories. Now, Are you ready for the answer? So the crowd answer is 20 to 40%. His actual performance was 96% correct in this task. In one of the blocks, he got 100% correct on the task. And so, outperforming the majority of the young adult subjects performing exactly the same task is this individual over here. So I think that's a good highlight of just how much variability there can be in performance with age, and how poorly at some level, in some very fundamental level, we understand the relationship between the changes in the brain that happen with aging and the corresponding changes in cognitive performance. And so, we're all, if we're tuning in, I think everyone is interested in this question: what are the brain mechanisms that support memory resilience in aging? And I'll kind of put forward the idea that, we can learn a lot from cross-sectional and longitudinal studies where we try to track people's changes in brain structure and function across the lifespan and try to relate those changes to whether or not they decline versus stay relatively preserved. But at some level, we'll have to go beyond this if we want really a mechanistic understanding, because cross-sectional and longitudinal studies can at best yield correlational evidence for brain changes that correspond to decline versus preservation. And if we want to further study mechanisms, I propose that one thing we could try to do is to use network-targeted brain stimulation, which I'll describe, as a potential method for more directly testing these mechanisms. And the reason is, this is because this method can be used, I think, to manipulate hippocampal network activity and thereby directly test hypotheses about how functional changes of the hippocampal network are responsible for memory decline versus preservation. So I'm going to start off with an overview of the network that supports episodic memory. So I think we should all be familiar with the idea that damage to the hippocampus, like in patient HM, causes a episodic memory disruption, the amnestic syndrome. And so the hippocampus is clearly an important structure for episodic memory. But it's important to remember that the hippocampus is situated within a broader network that accomplishes this function. And so the structure of this network has been fairly well articulated for quite a long time. So there's the hippocampus as well as a variety of cortical and subcortical regions with which it communicates. And so the anatomy was worked out quite a while ago, and this particular anatomy provides some insights about how the function of memory is accomplished. And so, to use the words of Marcel Mechelen, who summarized this anatomy and made some hypotheses about what the particular connected architecture means for mechanisms of memory ability, he said, "Behavior is not contained in the neuron or in the anatomical site, but in grids of connectivity that are both localized and distributed." The idea being that the hippocampus isn't accomplishing memory by itself, but it's really by virtue of the fact of its interactivity with this broader network that it's able to accomplish the complex function of memory. And so, as I said, Mechelen and others spent good parts of their early careers mapping out the structural connectivity of the episodic memory network as well as other networks. More recently, with the advent of methods such as MRI, we've been able to do things like map the functional connectivity of this network. So here's an earlier study by Randy Buckner and colleagues where they used resting state fMRI connectivity, they planted seeds in the hippocampus, and they identified areas around the brain that showed activity that correlated with the hippocampus over time while people rested in the fMRI scanner. And they identified this broad hippocampal network that maps fairly nicely onto the known structural connectivity network from the primate brain, and includes a distributed set of cortical and subcortical regions that presumably interact with the hippocampus to accomplish its cognitive functions. You can also consider this network functionally in terms of its participation in memory abilities. So for instance, here is a particular study from Mick Rugg and colleagues where they measured task-based functional connectivity. So these are areas that correlate with the hippocampus and their activity more so during recollection memory processing than during control task processing. Um, and what's color coded right here are consistency across different studies that use different methods of testing memory ability. And so the areas in red are those areas that are highly consistent across multiple independent studies in connecting with the hippocampus, particularly during periods of episodic memory performance. And so that's another way of functionally characterizing this broad network and showing its importance to memory. Indeed, it's been known for quite a long time that lesions of the hippocampus and kind of core areas of the classic PAPE circuit that are direct connectivity partners of the hippocampus cause the amnestic syndrome. This is some recent work by Mike Fox and colleagues where they really put an answer to the question of why do sometimes lesions that are distributed elsewhere throughout the brain also cause episodic memory impairment. And their answer was that these regions that cause amnesia, wherever they happen to be located, if they produce the amnestic syndrome, then they happen to be within the broad network of the posterior of the hippocampus and nearby areas of the mesial temporal lobe, like the retrosplenial cortex. So again, suggesting that it's not only lesions of the hippocampus that cause amnesia, like in patient HM, but really it's lesions of just about anywhere in the network of the hippocampus that cause the amnestic syndrome, and therefore, reinforcing the idea that really it's the hippocampal network that's critical for episodic memory ability. So the premise of what I'll talk about next in terms of our transcranial magnetic stimulation work is that if this network could be manipulated, then that would allow us to test how age-related decline and resilience are related to things like hippocampal network connectivity. So there's a literature that indicating that connectivity, resting state connectivity of the hippocampus and within the hippocampal network changes with aging, and some of these changes decreases in the network of the cortical regions of the hippocampal network, as well as some increases in connectivity of the hippocampus with itself bilaterally, are related to memory decline, for instance. Other ideas concerning the role of hippocampal evoked activity during memory processing. So there's some data to suggest that relative to young people, older adults that maintain their brain health have relatively similar higher levels of evoked activity while they're laying down new memories, versus older adults whose brains are declining structurally and have general cognitive decline, end up showing reduced activity evoked by this network during the formation of new memories. And so furthermore, there's some ideas that as the hippocampal network and hippocampal processing decline, you need to recruit additional areas in order to compensate for those losses. And so there are some reports of so-called compensatory recruitment, whereby in younger adults, you might get some areas of brain regions other than the hippocampus activated during memory tasks. In low-performing older adults, you get the same sort of regions as in younger adults, but in high-performing older adults who maintain their function, you get these additional extra recruitments that could be compensatory. And so if the idea is that if we could actually manipulate the network, that we could test these relationships in a direct sense by changing connectivity to see if that actually changes the corresponding memory ability that supposedly depends on that decline in connectivity, changing the evoked activity to see if that actually causes corresponding changes in performance, and so on. And so the first study that we did in this area, this is a study in healthy young adults, to demonstrate the general idea of manipulating the hippocampal network. So what we did is we took the hippocampal network as defined by resting state fMRI, and we applied non-invasive brain stimulation to an accessible cortical area of this network up over here in parietal cortex to answer the question, when we stimulate this cortical area, what happens to the entire network and how does that relate to memory ability? We used TMS which is this device right here. It's a very powerful electromagnet that you can hold up to someone's head and induce electrical activity in the superficial cortical neurons. To give you a demonstration of TMS working, it's not as though TMS passes electrical activity through the skull as what happens during methods like transcranial electrical stimulation, TDCS or TACS. It operates on the principle of electromagnetic induction, where a very powerful time-varying electromagnetic field magnetic field is turned on and off, that induces electrical activity in a perpendicular wire. Here it's an oscilloscope probe, and you can see every time the device clicks, you see a voltage induced in this oscilloscope probe. There's no lightning bolt shooting from here to here. So there's no actual electrical current passing from here to here. No electrical current has to pass through your skull to get into your brain. It's just induced at a distance by turning on the magnetic field on and off very rapidly. And you can generate very intense voltages on the cortical surface using this method. Of course, the electrical field falls off rapidly as a function of distance, and so you can really stimulate the cortex quite robustly with this sort of method. And you validate this in your individual subjects by aiming it at their motor cortex and causing contralateral muscle contractions, for instance, in the hand, in order to calibrate the device to make sure that you're at a sufficient intensity to be directly activating cortical neurons. So we use this TMS device and aim at this particular parietal area, like you're seeing in that electrical field plot right up here. And so in our first study, what we did is we defined the hippocampal network in each individual subject. So we put a target based off of anatomy in the left hippocampus of the subjects. We used resting state fMRI to define the network of the hippocampus, and we identified some of these stimulation accessible hotspots in the lateral parietal cortex. Other areas, of course, are part of the network, but they're too deep to be sufficiently stimulated with conventional TMS. We aimed the TMS coil at these cortical areas, and we stimulated people using this paradigm that's highlighted over here on the right-hand side of the screen. So we measured network connectivity and memory performance at baseline, and then we gave people 5 consecutive days of high frequency, 20 hertz repetitive TMS for about a half an hour a day to this cortical spot of the network for 5 days straight. Then people went home, we brought them back a full 24 hours later, and we tested the after effects of that stimulation on connectivity and memory performance. To answer the question of how did receiving these 5 days of stimulation change network connectivity and change their memory ability, we also performed various sham conditions, including low intensity stimulation for 5 days, as well as full intensity stimulation, but to other brain areas like the motor strip that are not part of the hippocampal network. These are referred to as the sham conditions, using the same 5-day paradigm. Every subject in the experiments got stimulation and sham in 2 different weeks with a washout period and a counterbalanced order. And so what we found, the main finding in this study, is that stimulation relative to sham increased resting state functional connectivity of the hippocampus with the rest of its network. So what's colorized up here is the statistical value for the increase in resting state functional connectivity of the hippocampus with these regions. These are all part of the hippocampal network at baseline. So these are all regions that, to start off with, were relatively well coupled in their activity with the hippocampus. The effect of stimulation was to increase the magnitude of that coupling, such that they were more tightly coupled. When one goes up, the other one goes down in terms of their activity fluctuation over time. So an increase in resting state connectivity for stimulation relative to sham. This is measured at the 24-hour mark, so a full day after they experienced the last pulse of stimulation. Now, these effects are quite, striking in that they're robust and also very specific, affecting the hippocampal network and not really other areas of the brain. And so it's nice to note that an independent group, ran a replication study. So these are data from Mike Friedberg, a postdoc, and Eric Wasserman's lab at the NINDS Intramural Research Program. They essentially ran a replication study of our original study and broke the brain down into the specific network areas that we found connectivity changes in the first study, and looked at the effect size of the effect of stimulation versus sham on connectivity. They also reanalyzed our original data, found essentially what we did in the first study, and found slightly more robust effect sizes in their replication study of connectivity changes in exactly these areas of the network. So it's interesting to note that even though we're applying stimulation at the parietal cortex, those connectivity changes that I was showing you were relative to the hippocampal target. The hippocampus was changing in its connectivity with the network. And so what we wanted to try to do is to sort out, even though we're stimulating at parietal cortex, what is really the locus of the connectivity changes that's occurring in the brain? Is it really a parietal mediated kind of effect, or more of a hippocampal mediated sort of effect, because that's our indirect target? So what we did to address that is we sorted connectivity at baseline based off of how well a region was connected with the hippocampus to begin with. So what's being shown in this graph over here, colorization indicates pairwise connectivity change across a bunch of ROIs in the brain. So bright colors indicate that stimulation increased the pairwise connectivity of regions. Cool colors indicate that there was really no significant increase in pairwise connectivity of regions. And what you can see is that regions changed in their connectivity the closer they were to the hippocampus in connectivity space to begin with. Things that were relatively less well connected to the hippocampus at baseline did not change so much. If you form the same graph but sort it, instead of connectivity with the hippocampus at baseline, if you sort it by baseline connectivity with the parietal cortex area that was actually stimulated, you don't see this nice organization. And in fact, there's a significant difference in so the prediction of distance from the hippocampus and connectivity space to begin with significantly predicts the stimulation effect. And that was also true in the replication sample. And so the bottom line is that even though we're stimulating the parietal cortex, the main locus of the effect seems to be on more of the core elements of the hippocampal network, the hippocampus, and those areas that are very highly connected with it to begin with in the mesial temporal lobe. We also looked at memory performance in this task. So subjects did this kind of classic episodic memory task where they study faces arbitrarily paired with words, and they later on see a face and have to recall the word. And you can see that stimulation improved their performance on this task, whereas sham did not, with a significant interaction between stimulation and sham. And notably, this task was given at the 24-hour testing mark. And so it's not as though subjects studied faces and then received 5 days of stimulation, and then we tested them on their retention of those faces that they studied before they received any stimulation. They had to learn the face name associations 24 hours after receiving stimulation or after receiving sham using different sets of stimuli. And the effect of stimulation is on the ability to perform this task, encoding and retrieval, 24 hours after stimulation was presented. And this memory enhancement effect also replicated in an independent sample, which you can check out if you're interested. Now, I'm not going to go too much into the various things we've been doing with network targeted stimulation in healthy young adults. The only thing I'm going to highlight is that across many different experiments, if you look at the effect size of stimulation relative to sham, for the effect on recollection memory ability due to this network targeted stimulation approach, what you see across a variety of studies, including some from other groups, is that the recollection memory enhancement is sort of in the medium to large effect size range. So fairly consistent improvements in recollection memory ability based off of network targeted stimulation. And these last for 24 hours and more for these 5-day stimulation paradigms that I've been describing. But the effects are a bit more short-lived in some studies where you use only one session of stimulation and you look just in the immediate hour after stimulation was applied. Either way, a memory enhancement effect, not a disruption. And these have been identified for stimulation of this parietal node of the hippocampal network without any changes in recollection memory for a variety of control stimulation locations, which I've cartooned over here. What I'm going to go into next is one of these particular studies, and this was a study where we used a very similar method, but in older adults. And so the experiment that we performed follows the same general setup and logic of the experiment I just described to you in healthy young adults. In older adults with no known neurodegenerative disorder, so they didn't have MCI, they didn't have AD, they were healthy older adults between 65 and 85 years old. They were screened for MCI and other disorders supervised by Dr. Sandy Weintraub at Northwestern's Alzheimer's Disease Center. So we're pretty confident they're relatively healthy, cognitively healthy older adults. They went through the same 5-day stimulation procedure as we ran in the young adults, where we identified the network, stimulated it for 5 days in a row, measured the effects of stimulation 24 hours later. We also did a 1-week follow-up. I'm mainly going to focus on the 24-hour effects in this talk. And the main question was, how does this network-targeted stimulation approach influence the way in which the hippocampal network supports episodic memory in older adults? And so to assess this, we used a task during fMRI scanning. This was the main outcome measure. And in this task, we had subjects study a form of associative memory. So they had to study in one set of blocks objects located at particular locations on the screen. So in this 6-point grid, so the school bus goes right here. In other blocks, they memorized that objects were associated with particular scenes. And later on, they had to take a memory test where they had to recognize the objects, say whether they're old or new. And then following each item recognition trial, they would make a contextual recollection judgment, where in this condition, they'd have to try to put it back into its original studied location. And in this condition, they'd have to pick the scene that it was paired with during encoding. Now, this contextual recollection component is the aspect of task performance that's particularly difficult and is particularly related to the hippocampus and should be the most sensitive to age-related cognitive decline relative to item recognition, which is relatively less dependent on the hippocampus and is relatively preserved in aging. And so the main question in the study was, what was the effect of stimulation on these different aspects of performance and their neural correlates? And so what we found is that stimulation improved performance on the contextual recollection aspect of the task most significantly. So there was a small but significant increase stimulation change relative to sham change for item recognition. They got a little bit better, but a marked improvement on the recollection aspect of the task, significantly greater than the improvement they showed for item recognition. If you want to break that out just for the recollection change to see how stimulation is a change, this is stimulation versus sham. Over here is plotted stimulation separately from sham, so you can see that this is being driven by an improvement pre to post stimulation for recollection in the stimulation condition, and no real considerable change at all in the sham condition. And so these are healthy older adults, but nonetheless, they're impaired relative to younger adults when they start off before they receive any stimulation at all. So if you just look at recollection at baseline, older adults are significantly impaired relative to younger adults performing exactly the same task. So again, these are 65 to 85-year-olds. Younger adults in this sample are 20 to 35-year-olds. And so there's a significant reduction in their ability to perform the recollection component of the task to begin with. And the question to kind of get a sense of what is the magnitude of the benefit that they see, that they experience from stimulation when they're stimulated, where does their recollection wind up relative to young adults? The answer is that it improves them, their recollection, almost to the same level as young adults performing the same task, with there no longer being a significant difference between old and young in task performance after older adults receive the 5 days of network-targeted stimulation. And this is, again, measured at the 24-hour mark after receiving 5 days of stimulation. So now the recollection is improving, and now the main question is, well, how is this improvement being supported by changes to the hippocampus and to the hippocampal cortical network? And so what we did to assess this is we got a neural correlate of memory formation within this task. So the encoding phase was scanned, and we subdivided trials according to pick out just those trials where later on they correctly recollected the contextual information that was paired with the stimulus at encoding. So only the correct recollection trials. And we asked the question, how does encoding related activity for these correct recollection trials change as a function of stimulation? How is stimulation changing the process by which a recollective memory is formed effectively? And we looked within the hippocampus to start. And so we broke the hippocampus up along its long axis. If you remember, we picked a particular spot, a target, based off of anatomy in the left hippocampus, and that's what we used as the seed to define the network that we stimulated. So this was really our target spot. And then we also looked anterior to our target spot along the hippocampal long axis and posterior to the target spot. Now, what did we expect to find? So there's some evidence to suggest that older adults who maintain their ability maintain high levels of evoked activity during episodic memory formation. So the old maintainers will have similar levels of relatively high activity while encoding memories, relative to older adults who decline and have relatively less evoked activity. So they're kind of hypoactive at some level relative to young adults and relative to older adults who decline. So based off of this sort of evidence, you might think that in order to enhance recollection, stimulation must have boosted up the level of activity in the hippocampus in order to go along and push recollection memory closer to that in young adults. Contrary-wise, there's quite a lot of evidence to suggest the opposite, that in fact, that poor performance in older adults is associated with so-called hyperactivity. So this is another study where they finding that relatively low-performing older adults, relative to higher-performing older adults, show relatively increased activation during memory processing in the hippocampus, as well as some of the regions of the distributed hippocampal network, not just in the hippocampus. And so this would suggest that increasing activity could be bad because that would be equivalent to hyperexcitability. These are both in healthy samples, not MCI subjects. And so really the question is, what did stimulation do to hippocampal activity going along with the increase in recollection? And so here's the time for our second poll, where you get to weigh in on what you think would have happened. If you've read this article or seen me talk about it before, please don't vote because that is biased. But if you haven't, please weigh in on whether or not you think that hippocampal activity during memory formation for these recollection trials stayed the same, no relationship, increased, or decreased.
So Joe, we will give it a few more seconds. About half of the audience voted.
Okay.
Maybe it's a difficult question.
Or maybe they've already seen it, so they
True.
they could have already read it, and they're being good citizens.
Okay, everybody, last chance. Okay, let's end it.
Okay, what do we got?
And here are the results.
Oh, so good. 69% of responders suggested that predicted there would be an increase. And so at some level, that means that I guess we didn't have to perform the experiment because you already knew the answer. So at any rate, so we did find relative activity increases for these recollection trials during encoding. And so, and we found them, so this particular segment right here was the indirect target. And so we found relative to sham stimulation increase evoked activity for these recollection trials during encoding at the exact area of the left hippocampus that we targeted, as well as more posterior regions of the hippocampus. This is actually consistent with our young adult data across multiple studies that suggest that really the posterior hippocampus is responding to the stimulation regimen more than the anterior hippocampus. At any rate, it hit our target as well as more posterior regions and increased their activity during recollection memory formation. This was specific to the left hippocampus. There was no significant change in the right hippocampus, which is the non-targeted side, so it's kind of serving as an internal control for the experiment. We also looked more broadly at the network. And so we used ROIs from a previous study by Randy Buckner and colleagues. These are all areas of the default mode network, which is, has a lot of overlap with the hippocampal network. These are all areas that show age-related connectivity decreases. And so we use these as ROIs to look at evoked activity, again, during recollection memory formation within them. We also used a control set of ROIs from the same previous study, areas that are not in the hippocampal network. And the question was, what did stimulation do to activity during encoding in these two different networks? And you can see that there were fairly consistent increases in activity across the individual ROIs of the hippocampal, of the distributed hippocampal network, with no such consistent increase in the distributed areas of this control network whatsoever. And if you look, one of the metrics that we performed is to see, it looks like a lot of the regions are changing and increasing their activity within this network. We ran an analysis, a correlational analysis, to answer the question, if within a subject, one ROI within the network changes, what is the magnitude of increase of all of the other areas? So essentially, how correlated within subjects are the increases across regions of the network? And what you can see, we call this coherence change, within the targeted network, the changes are very coherent. So if a subject shows an increase in one area, they also show an increase in the other areas. Whereas within the control network, any changes due to stimulation were not coherent. And at some level, the stimulation kind of decoupled the targeted network from the non-targeted network by increasing the coherence of activity within the targeted network relative to within the control network. So coherently increased activity across the hippocampal network as a function of stimulation for those recollection trials. A different way of thinking about the network is to consider its structure. And there's a good amount of data that aging is associated with cortical thinning of this distributed network, a lot of in regions that overlap quite considerably with the broader hippocampal network. And so I'm highlighting this particular study because in this study they found age-related cortical thinning in a set of regions that were related to hippocampal activity measured during task. And so for this is more evidence in sort of the hyperactivation is bad category. In these subjects, which included both healthy older adults and MCI subjects, more cortical thinning was associated with more hyperactivation that was negatively related to memory performance. And so the thinner your cortex in these areas of the hippocampal network, the more disruptively hyperactive your hippocampus is. And so the question that we had in this analysis is, if this hippocampal hyperactivation is problematic and related to cortical thinning of the network, then was stimulation, which increased hippocampal excitability further, detrimental for the subjects with relatively thinner cortex? So to answer this, we characterized cortical thinning in our group of subjects by comparing them to younger adults. And we found a very typical pattern of thinner cortex in areas that have shown thinner cortex associated with healthy aging, not AD, in a variety of previous studies. And so we looked within these areas to see what is the relationship between cortical thickness and how much improvement subjects experience due to stimulation. And we found, contrary to this idea that you would expect based off of the sort of previous evidence, so it was actually subjects with a thinner cortex that benefited the most from stimulation. And it was not because that subjects with thinner cortex had the worst baseline MoCA, age, or baseline task performance, so not just like a regression to the mean kind of effect. These variables were not predictors of the recollection benefit that subjects experienced from stimulation, whereas cortical thickness was. Thinner cortex being associated with a greater magnitude of improvement in recollection due to stimulation, with subjects having relatively very high thickness levels being the ones that really experienced essentially no benefit from stimulation in our study. And so to summarize these data, we found relatively durable 24-hour lasting rescue of recollection memory ability. This was associated with elevated activity in the hippocampus and its associated network, suggesting that this elevated activity is beneficial, not detrimental to episodic memory in healthy older adults. And that recollection benefits were greatest for older brains in the sense of thinner cortex, counter to the expectation that cortical thinning is associated with detrimental hyperactivation. So I think this is interesting and informative with respect to mechanisms of episodic memory change in aging, but a few caveats. So for one, I'd like to highlight the challenge of doing this kind of experiment. So if your interest is in running these kinds of studies in order to test interesting mechanistic questions about age-related decline and preservation of function, it's notable that running these experiments is quite difficult. So we started off with a sample of over 345 people who are interested in participating in our experiment, and we whittled them down based off of various TMS and fMRI contraindications to get our final very small sample. Another place where people fell where we lost a lot of people due to our phone screening was when they realized that participating in the experiment would involve coming into our laboratory every single day for 2 weeks straight, separated over the course of various months. I won't do a poll right now, but raise your hand if you want to drive into downtown Chicago every day for 2 weeks straight to be in an experiment. Not many people will raise their hand for that one. So it's quite challenging to get people to participate in these kinds of experiments. Furthermore, although I think they provide an interesting mechanistic leverage that is lacking in correlational kinds of designs, there are some limitations. So clinically, it's interesting and maybe promising that the memory benefits that we produce last for such a long period of time after we deliver stimulation. So, causing a relative rescue of memory performance out to 24 hours, I think is quite remarkable. But from a mechanistic standpoint, that's also a limitation. For instance, we administer the whole memory task at the 24-hour mark out here, and so we can measure fMRI activity at encoding and know that we're affecting it, but we can't really dissociate at a mechanistic level effects of stimulation on encoding versus on retrieval, or to get at whether they're mediated by transient effects on neuroactivity or more state-related changes that you would need to be able to go into. To do that, you need to be able to go in and manipulate activity of the hippocampus and related regions on individual trials. And so what I'm going to do in the last bit of the talk for just a few minutes is to quickly overview some of the studies that we've been running with healthy young adults, where we've tried to increase the methodological ability to conduct such studies, which might ultimately be yet more informative mechanistically. And so all the studies I've been describing, we went after the hippocampal network based off of its connectivity. One of the motivating focuses of the study I'm going to talk about is that we might be able to cause a more immediate and direct impact, i.e. on the single trial, as opposed to long-lasting changes over longer periods of time, if we could not only tailor stimulation to the specific location, but also use other properties of the network in order to tailor stimulation in order to have an immediate impact. And for these studies, we've been considering the idea that the hippocampus has a particular activity rhythm that can be measured, the theta nested gamma activity rhythm, whereby there are bursts of gamma activity in a theta rhythm. This can be measured in the hippocampus of rodents quite predominantly, but it's also there in humans, and in particularly synchrony among regions of the hippocampal network. These are data from Brett Foster's lab, using intercortical recordings from patients undergoing neurosurgeries. The areas of the hippocampal network couple at theta frequencies, suggesting that this might be a particularly good rhythm for communication among regions of the hippocampal network. And so what we've been doing is we've been trying to create trial level modulation of hippocampal function using theta rhythmic stimulation applied while people are actually being scanned in order to confirm that a single train of stimulus can have an immediate impact on hippocampal activity during memory task performance. And so for the one experiment we performed using this method so far, the brief overview is that we gave people different kinds of stimuli to look at while they were being scanned, either scenes, which the hippocampus really loves and gets very activated to, or numeric digits, to which people had to make very boring odd-even judgments, and the hippocampus doesn't really activate to these things at all. Each individual stimulus was preceded by 2 seconds of some stimulation condition, either on or off, and the stimulation conditions included the theta rhythm that we think the hippocampus should really listen to, versus a control frequency, a low beta rhythm that we do not think the hippocampus should really listen to. And we also applied these rhythms either to a parietal area targeting the network or to a control area in the supplementary motor cortex. The hypothesis is if we're able to have an immediate impact of the rhythm of choice on entraining the theta rhythms of the hippocampal network, then we should put those rhythms in the hippocampus, the theta rhythms, into alignment. And when they see a stimulus that they like to respond to, their responses will therefore be temporally aligned and create the largest evoked response because of that temporal alignment. Whereas even if they're aligned, they won't respond to things they don't care about, like these numeric digits. And if there's no effect on the rhythm, then the responses to the stimuli, they'll still be responded to, but those responses will not be temporally aligned, and they still won't respond to numeric digits. And so that's what we tested. We looked at these different conditions in the left hippocampus, which is what we targeted, again, based off of its connectivity with the stimulation location. The hippocampus did not really respond to numeric digits, and it didn't matter which stimulation condition we applied. In contrast, there was a response to scenes, and in particular, when stimulation was applied for those 2 seconds using the theta burst rhythm targeting the network, that created a significant increase in the evoked response relative to all other stimulation conditions. So the hippocampus immediately listens to that theta rhythm when applied for only 2 seconds. And there was a numeric trend for a similar effect in the right hippocampus that was not significant, kind of serving as a within-brain control for our targeting. And memory was improved in a corresponding way. And so recollection of the scenes was increased for the theta burst condition applied targeting the hippocampal network, not for the other stimulation conditions. So this suggests that it might be possible to have trial-specific effects on hippocampal memory processing if you use appropriate stimulation parameters targeting the appropriate locations and at rhythms that the brain regions that you care about like to listen to. And so to summarize everything, I think that network-targeted brain stimulation can be useful in testing mechanisms for cognitive decline and resilience, at least in the case of episodic memory. Memory enhancement in older adults via network-targeted stimulation was associated with some surprising network functional changes, activity increases, and no hyperactivation cost. Suggesting that age-related reductions in network activity are part of the causal pathway for memory impairment with age. And further insights into mechanisms could be gained via continued refinement of the non-invasive network targeting specificity through refinement of approaches such as concurrent TMS, fMRI, and others that can give you heightened ability to manipulate specific task periods and functions. And so with that, I'd like to thank my laboratory. These studies are a Herculean effort and take tons of people to accomplish. And so just about everyone in this picture made critical contributions to all the studies that I described today. Our collaborators on the studies, as well as the agencies that have provided funding for the research I described. And thanks again, Michael and others, for inviting me and for organizing this, and to everybody who tuned in.
Thank you, Joel. That was really a fantastic talk, and it's great to see that you could replicate your results across different samples. So it's a really robust finding, and you have really developed a tool that allows to manipulate a specific network, giving mechanistic insights. So now we have time for questions. Please come forward, unmute yourself, activate your camera. And yeah, so if anybody would like to ask anything, please use the opportunity now. while you may think about a question, let me start. Joel, so it's really intriguing to see that you found an increase rather than a decrease. But do you have an idea of how to, you know, in the hippocampus connectivity and also activation? But could you give us an idea of how to reconcile this idea of efficiency versus a deficiency in activation, which you may have manipulated? So based on the efficiency hypothesis, you know, you would have expected a decrease, and you know, often this has been observed, but you found actually that an increase was beneficial.
Yeah, it's a good question, and I think, you know, I would shy away from saying, you know, that, you know, the only way to study these things is by, you know, network targeted TMS. You know, that's like an overstatement. But you know, I think, you know, thinking about, and I don't think my study provides an answer to that question, you know, in any way.
Mm.
But I think it could be a useful tool, because you could take, you know, there are been a variety of studies that are in conflict of whether or not they see evoked activity increases versus decreases, and oftentimes those things are argued away as being differences in, you know, in the sample that happens to be included, in their particular cognitive or clinical variables. So for instance, there's some idea that, you know, as people start to have some pathology and transition into the MCI phase, maybe now that's when excitability becomes pathological. Whereas before then, excitability, relatively healthy levels of excitability is good, you know, and differences in the tasks that are used across the different studies that have been used to measure it, and all these other variables. And so I don't think my study can address the question exactly, but if you think about it as being a tool where you could take the same sample and the same individuals and hold them and their brain structure and the task testing, you know, situation constant, and then run a manipulation like this, then you could actually get a better sense and test, you know, the directionality and of the relationship between evoked activity and performance of the task, holding all other things constant.
Right. Yeah.
So I don't have an answer. You know, I you know, went into it being a little bit more familiar with work by John Disterhoff and the kind of healthy aging literature suggesting that, you know, relative decreases in excitability and task evoked activity are problematic, and that by restoring them in the rodents, by enhancing excitability, you can restore some function.
Okay.
But you're right, there's just as robust of a literature on the opposite side of things. So it's hard to say.
Okay, thanks. Anybody else?
Hi there. If I may, so I'm Till Bergman from Mainz, Germany. Thanks, Michael, for organizing this and making it open to the public. I really enjoyed your talk, Joel. Thanks a lot. So my question is more a conceptual one regarding all the TMS studies that try to indirectly target deep brain areas, right? Especially when you try to employ plastic effects, so induce long-lasting effects with RTMS. Where do you think do the RTMS effects actually take place? I mean, where is this synaptic plasticity happening? I know it's a network effect, right? But the deep relevant target is the hippocampus. So is the RTMS changing excitability of the parietal cortex of the entry point, basically, so the superficial side, which is then indirectly by its increased activity having an impact on the hippocampus as an after effect? Or is the after effect actually happening in the hippocampus itself, because the stimulation protocol induced repetitive remote effects like spreading into the hippocampus?
Yeah, I you know, I'd say that with fMRI, it's a little bit difficult to sort out that with much certainty. What I try to do, and I don't know what will happen if I stop. Maybe I've never tried zooming back to my previous slides while giving a virtual talk. I wonder if I can do it. Just a second. We tried to address that with fMRI to the best of our imaginations via this analysis, where we said we broke up the brain into a couple, about 100 ROIs on anatomical grounds, and we said what is the effect of stimulation on their pairwise connectivity, and like I said during the talk, we sorted them based off of how close they were to the hippocampus to begin with, right? And you see this nice organization where areas that are more well connected with the hippocampus before you get any stimulation at all, those are the ones that respond in increasing their connectivity, whereas you don't see the same thing with the parietal cortex, the site where stimulation is actually applied. If you sort it based off of its baseline connectivity with the parietal cortex, that organization does not come out. So it suggests that even though we're stimulating the parietal cortex, the locus of the plasticity is at somewhere closer to the hippocampus. I wouldn't want to say with much confidence whether it's the hippocampus or somewhere nearby or anything, but that's the best we could think to do with fMRI. The other kind of notable thing is that if you look across a variety of our studies at task-evoked activity changes as well as connectivity changes, they're really not robust at the stimulation site at the 24-hour mark, almost as though the stimulation site is sort of providing, at least in our hands, and with this particular network, seems to be providing more of a window to the hippocampus at some level, and itself, even though it's part of the hippocampal network, it's a relatively weak part compared to the more medial areas that have very strong connectivity. And those are the areas that typically have robust lingering effects at the 24-hour mark, not really the area that we're stimulating. And so the kind of conceptual model we've been going off of is that high frequency stimulation will at some level pass to the hippocampus, through transsynaptically and create perhaps a LTP-like effect, which is associated with network level potentiation. So there are some neat data from the Logothetis lab that if you just induce hippocampal LTP, you get an effective connectivity change throughout the network. This is measured in rodents using direct electrophysiological methods to suggest that it could be an important locus for mediating network-wide kind of plasticity, perhaps. But we don't really know for sure. We have just started a 5-year collaborative project funded by the NINDS with John Disterhoff's lab at Northwestern, where we've developed a rodent model of the stimulation approach. And so we're hitting cortical network spots in the rodent, and we're going to try to sort that question out by doing in vivo and in vitro hippocampal electrophysiology to try to address that. But fMRI, very good point. Yeah.
Thank you. I think we have another question.
Yeah, so you showed some frontal activation from Roberto Cabeza's work, and this is a focus on compensation. So I was wondering if you found any effects on the frontal lobe when you enhance hippocampal, so you don't. And then also you showed a study where you had different seeds within the hippocampus, and I was wondering if you looked at areas within the hippocampus, anterior versus posterior, whether there were any interesting subdivision effects.
Yeah, so so with the first question, I kind of gave a quick, maybe not very sufficient response, but you're right. We thought that if those prefrontal regions are compensating, then you'd expect them to no longer need to compensate if you enhance the hippocampus and therefore show a relative reduction in activity. We didn't see that, but the caveat is that the experiment we ran wasn't really geared towards dividing the people who are probably compensating from the people who are not, like in Roberto's studies, where they carefully try to say, okay, well, who are the subjects who maintain their performance level and show the compensation. We only had the sample size relatively small, 16, to look at the group level, and we don't see any group level PFC activity changes as the hippocampal network goes up. But that's a kind of a neat idea. I think you could imagine using this to more carefully test that hypothesis with the proper subdivision of subjects. And then the second question was whether or not there is any differentiation within the hippocampus in terms of what responds. So yes, in all of the 5-day stimulation studies that we've run, the effects seem to be more robust for the posterior hippocampus, which is consistent to some degree with the anatomy. So the parietal cortex projects more into retrosplenial and parahippocampal cortex than it does pararhinal cortex. And so it's, and those are the, and so the posterior hippocampus is more what we see responding. We've never tried doing any sort of subfield analysis or anything like that because I think our resolution is a bit insufficient, but
Thanks.
yeah.
Okay, we have another question posted via text. So the question relates to real-world evidence of memory changes. Did you ask or did you assess whether subjects had actually noticed a difference in their everyday memory abilities?
Yeah, we did. We gave people a couple of subjective questionnaires. One was the everyday memory questionnaire. And we did not see an effect of stimulation on the everyday memory questionnaire. I think it's a little bit challenging to know what that means exactly because a lot of the questions concern very specific instances of memory activities that the subjects may or may not have actually done in the period between when we stimulated them and when we gave them the questionnaire. So for instance, remembering people's telephone numbers. I don't remember the last time I've ever had to actually remember someone's telephone number instead of just hitting it on my phone, and things like that. And, some activities that were assessed, they simply might not have done. And so it might not have the sensitivity to pick up on those subjective kinds of things. People, older adults in the study, some of them reported feeling more alert and other kinds of subjective states, but nothing really systematically. And we didn't pick up anything on the everyday memory questionnaire. So.
Thank you.
Yeah.
Any other questions?
I have one. Thank you for the talk. So maybe you say that, but I missed. So you only stimulate the left parietal cortex. Is that right? So why you focus only on the left hemisphere, if there is some reason? Thank you.
Yeah, no problem. I really wish that I had a very wonderful theoretical rationale for focusing on the left. And you could make an argument that the left hemisphere might, it has slightly more, memory related function perhaps than the right. And there's some evidence for that. But to be truthful, we focused on the left in our very first study because we did. It was sort of by happenstance. And we haven't, for the most part, had the resources or time to expand everything into the